So the first topic today is about graphing, setting up a scientific graph. Here's a good example. On your x-axis, you always put your independent variable. That is the thing that you are manipulating. You've got to include a unit. Make sure that's there. On the y-axis, you're putting your dependent variable, which is the thing you're actually measuring. Something that's not on this one that you always want to include is a title. And the surefire way to get credit on the AP exam for giving this a title is say something like the effect of your independent variable, in this case time, on your dependent variable, in this case milliliters of oxygen consumed. Again, make sure for both your variables you have units. If you're graphing points, and let's say this was actually our first point and this was our last point. In this graph, they didn't really do things correctly because they went past their actual data points. If you extend your line past your data point, how are you supposed to let the person who's looking at your graph know that that's just a guess? Dash it or dot it. Don't keep it as a straight line. Let them know that this little segment right here, you don't really have data to back that up. You're guessing. Same up here. It's a guess. And you need to let them know that with like a dash line. Something else you're always going to have to do on these things is calculate rate of change. In any graph, key point, rate is slope, which you guys should know is change in y over change in x. So let's say we wanted to know the rate of oxygen consumption between 5 seconds and let's say 15 seconds for this line right here, germinating corn at 22 degrees Celsius. And we want to know the rate between 5 and 15 seconds. So at 5, our y coordinate is about 0.4. And at 15, our y coordinate is essentially 1.2. So if we're going to calculate the rate, change in y, 1.2 minus 0.4 over change in x, 15 minus 5. So we're up to 0.8 on top over 10 on the bottom. So our rate is 0.08. The number's not enough. You got to have a unit. It's the y unit, milliliters of oxygen, divided by the x unit, minutes. So milliliters of O2 per minute. And that should take care of it. So again, key, kind of summing that up. X-axis, independent variable with a unit. Y-axis, dependent variable with a unit. Give it a title. The effect of your independent variable on your dependent variable. If you happen to graph it, another point here while I'm thinking about it. If your actual data points aren't in an exact line, there's something like that. What are you supposed to draw in? A best fit line. Don't snake it. Draw in a best fit line. Now, if your data points look more like this, say these are your data points. That's obviously not a straight line. Don't try to best fit that. But if your points are something like this, that's linear. So draw in a best fit line. Here, that's a curve. That's a quadratic. Don't try to best fit that. The other thing, rate is slope. Change in y over change in x. All right, topic 2 today, neurons, nerve cells. Key thing to know about nerve cells, there's 3 main parts. You've got these dendrites up at the top. They look sort of like tree branches. They receive incoming information from sense receptors or from other nerves. In the middle here, you have the cell's nucleus. That area is called either the cell body, or a word that we've seen a lot in this class, soma is another name for body, like somatic cell. Looks like a hand. Coming out the other side is an axon. And that axon sends out outgoing information. And it's going to send out that outgoing information to either a muscle or to another nerve. This particular kind of nerve we're looking at here is a motor nerve, which means it's connected to a muscle. It's carrying information from the central nervous system to a muscle. Over here we're looking at a sense nerve. This sensory nerve is carrying information from a receptor to the central nervous system. And then interneurons we won't say much about today, but in like a reflex arc, they connect directly up sensory nerves to motor nerves. Key things to know about nerves. Nerve cells are normally polarized. What that means is that the way nerves are set up, they have a lot of sodium ions outside the nerve and a lot of potassium ions inside the nerve. There's a concentration gradient. The potassiums are not the only thing inside the nerve, though. There are also a lot of negative things in here, like proteins and chlorides and stuff. So inside the nerve has a net negative charge. Outside the nerve, there's a lot of positive sodiums. So we start off in a nerve with a voltage called the resting voltage of about negative 70 millivolts. When this nerve is stimulated by like a neurotransmitter or something, that causes certain gates on this nerve to open up. Gates mostly for sodium. And when those gates open up, sodium rushes in. That causes this voltage to shoot up. That's a current of moving electrons, or moving not electrons, but in this case of ions. And whenever ions move, that's electricity. So this is a nerve impulse. It's what we call an action potential. Sodiums move in. As the sodiums start to move in and the voltage goes up, that opens more and more channels. They're voltage-gated channels. So they move in faster and faster and faster and faster. And that's why this voltage shoots up so fast. But eventually we get it to the peak, and it's done what it needs to do. Positive 40 millivolts is what we call the peak voltage. When you get up there, other channels in this neuron open up. But these are channels for potassium. And since there's a lot of potassium in the nerve and not much outside, the potassium comes out. Potassium's positive too. So as those potassiums come out, the voltage goes back down. So we get down to the right voltage. We get down to negative 70. But when we get down here, the nerve's still not ready. Because for a nerve to work, it needs to be polarized. It needs to have sodium on the outside and potassium on the inside. And the thing that makes that happen, the thing that gets it back to where it needs to be, is one of the most important structures in your body. It's called the sodium-potassium pump. And its job in nerves is to take sodium from inside the cell, pump it out. To take potassium outside the cell, pump it in. And that helps to get that nerve back to that ready state, the polarized state. During this period of time right here in our action potential, that's what's going on. The sodium-potassium pump is getting the sodium back out here and the potassium back inside here. During this period of time, the nerve won't work. But that's not a very long period of time. That's a little bit over 1 millisecond. So it doesn't take long for it to recharge. This whole thing that I just described is called an action potential. And that is a fancy name. I'm just going to spell that. A fancy name for a nerve impulse. The electrical signals that travel through all the nerves. That right there is what makes you think, remember, feel, have emotions. Little electrical signals all through your body. Continuing on with nerves. When a nerve impulse travels down an axon, it eventually is going to get to the end of the axon. This area. When it gets there, a lot of people think that electricity just jumps across this gap to the next nerve. That's not what happens. When the signal gets to the end, the action potential gets to the end, calcium ions flow into this neuron. And they cause the neuron to basically spit out these little chemicals called neurotransmitters. Do exocytosis on these neurotransmitters. These little guys that we see down here. These are cell signals. They're ligands. These cell signals will cross this gap between two nerves, called the synapse, and they'll bind on the postsynaptic neuron to receptors, special receptors. Those receptors are called ligand-gated ion channels. And once enough of these neurotransmitters bind to these channels, receptors, it causes the channel to open. These are those initial sodium channels that start up the action potential that we just got through talking about. So this is the beginning of an action potential. And it's caused by neurotransmitters from one nerve opening up gates on another nerve, gates for sodium. There's a lot of different neurotransmitters in your body. You ought to recognize the names for a few of them. This may be the most important one, acetylcholine. It's involved in like nerves talking to muscles. Some you've probably heard of on TV before, dopamine and serotonin. And you've probably heard of those before on commercials for like antidepressants, like Prozac. Because when these two things get out of whack, you don't feel so high. Right, but know that those are neurotransmitters, and know that neurotransmitters are cell signals that let one nerve pass its impulse on to the next. And they do that by crossing a synapse, binding to a receptor, and then opening up sodium channels, which will lead to the creation of an action potential. All right, here we're looking at an energy pyramid, or what we might call a trophic pyramid. Always at the bottom, primary producers. Plants, algae, or bacteria. Cyanobacteria in particular. The things that eat them are primary consumers. The things that eat them are secondary consumers. The things that eat them are tertiary consumers. But you notice that the width of these blocks decreases as we go up the food chain. And that's because there's less available energy to each chain. The plants at the bottom can use the sunlight. But as they use it, some of that energy gets used up by the plants, turned into heat. A lot of it gets turned into heat and wasted. Only about 10% of the energy used by this level makes it to this level. The rest is lost. Only about 10% at the primary consumer level makes it to here. Only about 10% of this makes it to here. So as we go up a food chain, or up a trophic pyramid, there's less and less energy available, which means there's less numbers of organisms. So the amount of available energy regulates numbers. And that's what we see happen here. And we also see at the end of any food chain are microorganisms, decomposers, detrivores. If you hear that word detritus, that just means dead stuff that needs to be decomposed. Those things play an important role in what we see somewhere on this diagram. Where's it at? May not be written over here this time. Mineral recycling. They take minerals out of these dead organisms and put them back into the ground. Yeah, that's usually in here somewhere. But that's important for keeping the food chain going. Those minerals usually act as like cofactors or coenzymes to help keep things working. All right, here we're looking at pie charts that show how energy is used in different kinds of organisms. Human, penguin, and this is a deer mouse. It's not actually a deer, it's a deer mouse. All these are endotherms, warm-blooded organisms. And then we've got a snake over here, which is obviously ectotherm, cold-blooded. As you look at this pie chart, notice the blue is normal metabolism, the BMR, the basal metabolic rate. Red is for reproduction. Orange is for thermoregulation. And yellow is just for general activity levels. If you notice the basal metabolic rate is pretty big in all of them. But the part that we want to concentrate on is this orange part. Notice in the female human, that's a fairly good chunk of the pie. A little bit bigger chunk in the penguin, and way bigger in the deer mouse. Now all those are thermoregulators. They're all endotherms. Why does this little bitty mouse use so much more percent-wise of its energy than a human does to maintain body temperature? And the key to that answer is that when you're small, your ratio of surface area to volume is bigger. So when you're small, your ratio of surface area to volume increases. This thing has way more surface area per unit of volume than she does, which means she has way more places to lose what? To lose heat. And since per cell, this deer mouse has way more places to lose heat than this woman does, to maintain body temperature, the deer mouse's metabolic rate has to be much higher than the human females. An elephant per cell has a way lower metabolic rate than a mouse per cell for the same reason. As things get bigger, their surface area to volume ratio decreases. So they become more efficient. They become better able to maintain their body temperature. When you're small, your surface area to volume ratio is really big, which means there's lots of places to lose heat. And therefore, the only way to maintain body temperature is to generate a lot of body heat, burn a lot of sugar. And that's what we see going on here. Another thing to point out, the python doesn't have an orange piece because he's not a thermoregulator. He doesn't use body heat to regulate his temperature. He might use his behavior to regulate a temperature. He might go in a hole if it gets too cold. He might go in a cave if it gets too hot. He might lay on a rock and bathe in the sun to warm himself up. The brown is energy for growth. So they're not growing. None of these are growing a whole lot at this point in time. We're looking at adults for the most part. Key thing though, small things have a larger surface area to volume ratio than do big things, which means they have per cell more places to lose heat. And therefore, to maintain body temperature, their metabolic rate per cell has to be higher than bigger organisms like humans and whales and elephants and things. Next topic we want to look at, enzymes and metabolism. Here we're looking at what's called a metabolic pathway, a set of steps that make an ultimate end product. In this case, we're trying to make an amino acid, isoleucine. Isoleucine in your body can't be made in one step. It takes a lot of different steps to do it. We start off with an initial substrate called threonine. That's another amino acid. We have the first enzyme here in the pathway that takes that threonine and turns it into an intermediate. It changes it into something else. This intermediate then becomes the substrate for enzyme 2. Enzyme 2 takes whatever intermediate A is and turns it into B. A different enzyme takes B, turns it into C. So it's a chain of steps. Eventually, that chain of steps leads to what we're trying to make, isoleucine. That whole chain of chemical steps is a metabolic pathway. But here's the thing. You've got lots and lots of metabolic pathways in your body. You don't want them all working full speed all the time. You need them to be regulated. You need to be able to turn them off and turn them on. I've made this statement before. Your body is conservative by nature. Liberalism leads to what? Starvation and death. Okay? Your body is here trying to conserve its resources. So here's the thing. If you have enough isoleucine, it makes no sense to continue making more. So when you get up to a proper concentration, this pathway needs to turn itself off. And how that works typically, when the concentration gets to a certain level, the product of the pathway, in this case isoleucine, will bind to a little groove on the first enzyme in the pathway, or at least one of the early ones. And that little groove, sometimes it's called the feedback site. Sometimes you're going to see it called the allosteric site. Allosteric means that it's going to change what? The shape of this enzyme, and thereby turn it off. This process that I just described to you is called feedback inhibition. When the product of a metabolic pathway gets to a certain concentration, it will bind to the feedback or allosteric site of one of the early enzymes in the pathway, and thus turn it off. But it's not an eternal turn off. That product is going to stay binded to that enzyme until the concentration drops. And when it drops, it's going to turn loose, and the pathway will turn back on. This is conservatism. Make it until you got enough. Stop making it. Save your resources for later. All right, guys, another example of negative feedback we're going to look at right now. This is how we regulate our body temperature. All right, in terms of homeostasis, our body temperature ought to be between 36 and 38. 37 degrees Celsius is about the same as 98.6. Well, let's say that your body temperature starts to go up. Your hypothalamus is going to sense that, and it's going to say, that's not a good thing. So to keep your body temperature from going too high, your hypothalamus is going to send out messages, usually nervous, but it could be also hormonal, to your sweat glands. And it's going to say, make sweat, send that sweat to the surface of the skin, evaporate that sweat, lose heat. That's one response. Another response is to send messages to the blood vessels near the surface of your skin. And if you're getting too hot, the message is to take those blood vessels and dilate them, so that now there's more blood near the surface of your skin, which means it's easier for heat in that blood to do what? To leave. That's why people turn red when they get hot. It's a way to lose heat. So that starts to cool the body temperature off. If not cool it off too far, let's say that it does. Now our body temperature is dropping a little bit below 98.6, 37. So now our hypothalamus says, let's don't overdo it. Let's send out a message now to those blood vessels near the surface of the skin, and let's tell them to shrink in, constrict. Therefore, the blood gets kept mostly in the core, away from the outside, so it maintains heat. Also, it may send out messages to the muscles and tell them to start to shiver. That'll generate some friction and some heat. And something that's not up here, it may also send out a message to the endocrine gland that we call the thyroid. And the thyroid gland will release a hormone that will speed up what? Cell respiration, metabolism, which generates what? Heat. And that will warm us up even more. So this thing that we're looking at here is a negative feedback loop. Do something until you get back to normal, and then stop. Try to maintain homeostasis. So we've got a statement that I was trying to pull up here. Let's see if I can pull it up. So negative feedback, a change in the variable being monitored counteracts further change in the same direction. It's sort of a summary of what negative feedback is. You get whatever you're trying to keep in homeostasis to that level, and then you try to keep it there. If something changes it in one direction, you change it back to the other direction. You undo what your body does to get back to normal. Negative feedback. All right, last topic on this video, biotechnology. This is one of the labs that we have not done yet, that we will do, but the way it typically goes, that's the one they're going to ask about on the AP exam, so we need to make sure we know about it. An important term to know is recombinant DNA. There's all kinds of examples of this, but maybe the most famous one is human DNA, the human gene for insulin, has been inserted into bacteria. And you get a piece of DNA that's part bacterial, part human. That's recombinant DNA. It's a combination of DNA from two or more different things. Now the trick is, what we're trying to accomplish by doing this, if we can get this piece of DNA, this piece of recombinant DNA into a bacteria, then every time it divides, it's going to copy for us. And every time it does transcription and translation, it's going to make whatever this gene says to make, which is insulin. So this is how insulin for diabetics is made. It's made in big vats of millions of bacteria, all chugging out not bacterial insulin, but instead making human insulin because they're using directions from a human gene. Now to make that was a little tricky. One of the key things to understand in this process of recombinant DNA are these enzymes right here, restriction enzymes. These guys cut DNA after only specific sequences of A's, T's, G's, and C's, nucleotide sequences. And because of that, when you cut up a piece of DNA with these, oftentimes the piece of DNA gets cut in a crooked fashion. So that you have unpaired bases sticking out here. Like you might have an A and a T and a C and maybe another T. If you have another piece of DNA with the opposite bases, the complementary bases, like T, A, G, A, those pieces will stick together. And that allows us to make this stuff, recombinant DNA. So if you're trying to make a piece of recombinant DNA that's part human and part bacterial, you're going to cut the human DNA with a certain restriction enzyme. Then you're going to take a piece of bacterial DNA and cut it with the same one. If you use the same enzyme, these little, what we call sticky ends, will match up, and you can put them together. Once you do that, you make this connection permanent using the same enzyme that was used to put together the Okazaki fragments during replication, DNA ligase. And that will make that a permanently connected piece of recombinant DNA. The trick then is to get that piece of DNA into bacteria. You have to treat them with some chemicals. Sometimes you'll heat them up and cool them down to make that happen. But if you take a piece of DNA from one thing and you put it in something else and change it, that's called the transformation. Taking DNA from one source, putting it into an organism, and changing it. In this case, we're going to transform bacteria with human DNA. An example of this that you may have heard of before, a lot of crop plants, early spring crops that bloom like in March. A lot of times they'll get killed by frost. Scientists have found this gene in Arctic fish that makes a natural antifreeze. They took that gene out, it's a fish gene, and they put that fish gene into crop plants like peaches, strawberries. That gene codes for this antifreeze. So now these peach trees, even when it frosts, they keep on going because they have a fish gene in them. Now that grosses a lot of people out when they're eating peach, they're eating fish. Not really. They're eating one particular protein from a fish out of how many? Thousands. Okay, thousands. So you're not really eating fish. They're eating one fish protein, and it's making their peaches. It's making farmers be able to grow more peaches cheaper, which means you can buy them cheaper. So I think this is a good thing, not a bad thing. The other way that this gets used is, let's say that we took a piece of my DNA and we cut it up with these restriction enzymes. My DNA is going to get cut into chunks. We're going to have these chunks called RFLPs, restriction fragments. And then we took some of Emma's DNA. We cut it up with the same enzyme. Are my chunks and Emma's chunks going to be the same chunks? No. Because these enzymes cut after very specific sequences of A's, T's, G's, and C's. So my DNA and Emma's DNA is not the same. So we're going to get different fragments. We'll take that cut up DNA and put it into a machine called an electrophoresis chamber. That's going to expose it to electricity. It's going to cause those little fragments to move, and they're going to separate into bands. And that banding pattern that we get, that's a DNA fingerprint. And we know that can be used for criminal cases. It can also be used for paternity kinds of cases, like who's the daddy kinds of cases.
Is that process called?
No, electrolysis is something different. Electrolysis is splitting something using electricity. Electrophoresis, different things. Here's the situation with the paternity case. You got this baby over here, who Mary is the mother. Bob and Larry are potential fathers. Mary doesn't know which one the father is. They both could be. So what happened? Well, this is like an episode of the Maury Povich show, right? You've seen that show? So they're on national television. She admits that either one of these guys could be the father. The show pays to take all their DNA, cut it with restriction enzymes into fragments, run it through this electrophoresis chamber, separate it into bands. Compare the bands. Here's the way it works in paternity cases. Every band in this child has to be accounted for from the mother and the father. And actually it's 50-50. This diagram doesn't show all the bands. In actuality, it would be a lot of bands. But it's pretty obvious that this band is coming from mom. That's okay, just let it go. This band right here is coming from Larry. Bob does not have this band. Bob cannot be the father. This band right here is kind of interesting because Larry has it, and so does Bob. What might that mean about Bob and Larry? They might be kind of related, which is because Mary's not a very nice person, right? And then down here, this band also obviously comes from Larry. So Larry's the father. Every band in the child can be accounted for either from the mother or from Larry. Larry's the father. Bob might be the child's uncle or something. He's not the father because Bob and Mary together cannot account for all those bands.